My name is Astha Symes, and welcome to Live Longer World. My guest today is Dr. Alfonso Martinez Arias, who is a developmental biologist. He is also the author of a fabulous book called The Master Builder: How the New Science of the Cell is Rewriting the Story of Life. His point is that in biology, we focused too much on genes. We attribute every understanding of biology and disease states to genes, but that's not the case. Many of us know that genes don't have all the answers to problems in biology. So what if we should also be looking at cells and understanding their behavior? In today's conversation, we talk about the cell's eye view of biology and how cells might be the ones controlling genes, and how understanding cells gives us better insights into development, longevity, and the frontiers of biology. We also talk about the incompleteness of Richard Dawkins and how his theory of the selfish gene might in fact not be fully accurate. I hope you enjoy today's conversation. I read your book, The Master Builder, which is right here with me. And I think it's a fascinating book because you're making a very counterintuitive statement. And you're talking about the cell's eye view of biology. So one of the statements you say is, what makes you and me unique as individuals is not a unique set of DNA, but instead a unique organization of cells and their activities. This is not what we've been told. We've always been told it's our DNA that makes us unique. DNA has been ingrained in the narrative, even in common parlance. So why do you say it's not the DNA, but instead it's the cells that make us unique?
Well, sometimes the things that are staring at us, the obvious ones, are the ones that we tend to overlook. It's always surprised me that we seem to rely for our identity on what we cannot see, which is DNA, when what we are seeing is cells, it's not DNA. The reason that makes you the way I am seeing you in this video, and me, the one, is not our DNA. In fact, as I'm sure we'll discuss in the next few minutes, there is a very long way from the DNA to the cells. I mean, what the reason why you can see me is because there are cells working in your eyes that are taking up the information and firing through other cells to the brain that is made up of cells. Genes have very little to do with that. Okay, so I think that's one of the things. But then there is a lot of. I think the reason for all this is that the DNA is very easy to understand. You know, you have a sequence of A, G, C's, and T's. We can there is some kind of identity associated with the sequences, the billions of letters that we can arrange those 4 letters into or sentences, and therefore it looks like there is some reality to the notion that there is some connection between us and the DNA, but there is no more connection that there is between the barcode of a product in the supermarket and the product itself. The barcode is something that identifies that product, but it's not the product. The product contains a lot of other structural facilities. But then there are features in life that do say that clearly there is. It's not that the genes. I want to understand. I want to make this clear. It's not that the genes do not have anything to do with us. But it's certainly, as you say, it's not what gives us our identity, our shape. So, for example, maybe people that are listening to us have brothers or sisters, and we are different from each other. You are slightly different from your brother or your sister, and they are slightly different from you. One of you might be taller, one of you might be shorter. However, what would happen if we could combine both of you? Would you get a mixture of the two people, or would you get a person that is totally different? Well, this happens in nature many, many times. These are called chimeras. There is about 15% of the people moving around. It's people that have 2 DNAs within their body. Very early in their development, the very early embryos fuse into one person. And now you get a perfectly proportioned person, even though there are 2 totally different DNAs. Had those 2 very early embryos developed into individual people, they would have been different. However, they somehow negotiate And it's not the DNA that is doing that negotiation. It is the cells that are doing that negotiation. So I think that that's what I meant when I said that sometimes when it's obviously staring at us, we tend to forget. I think there is many reasons why there have been a push to find our roots into the DNA. Some of them are right. I'm not saying that DNA has nothing to do with us, but I do think that the cells are something that we need to explore.
So I want to dive deeper into something you said there, which is that the DNA is not doing the negotiation in terms of, say, the structure, but it's the cells. How do we know that? Why can't it be the DNA?
Because the DNA is responding to what's happening in the cell. So I want to make one thing clear, two things very clear from the beginning, because there is many people out there looking for headlines and looking for. There is some people that say, not many, but some. There are many that we don't hear to. There is a few that we listen to. Oh, everything about the genes is wrong. There is nothing in the genes, or. That's not the case. I'm not saying that. I'm trying to understand the relationship between the genes and the cells. I think we've learned a great deal about genes. Now, if you think about it, and my background has led me to think about that, we can explore that in a minute. I think you realize that cells, we know very little about cells. We need to know more. And that the little that we know tells us that they are using the genes. They are not a simple, direct product of the genes, but they are themselves using the genes in an interesting manner. And that becomes obvious when it is about to make individuals, to make people, to make, it's very, very clear. For example, we've done a number of experiments in the lab where, depending on the initial number of cells, you're going to make a structure that looks like an embryo, perfectly organized or not. If you have too few cells, nothing happens. If you have too many, a mess happens. But if you have the right number, all of a sudden you start getting structures that can organize themselves. In every case, it is the same genes. It is the same cells, but obviously the cells are able to sense their number, their organization, and now they're going to be using the genes to start building things. All right? It's what we said, when these two embryos that give rise to a chimera, and there are many examples. I discuss one particular famous one in the book. When two embryos come together, there are two different genomes that, as we said, had they developed independently, they would have given rise to two different people. But as they give rise to one person. So I think those are examples that it is the cells that are always looking around, processing information, if you want to use that terminology, that allows us to think, and then deeping into the genome to turn on and off genes. A very common metaphor that now it usually happens that when things become obvious, people start saying, no, we never said that. They said there is a blueprint. The genome is a blueprint for us. It is the recipe to make a human being. Now that it's becoming obvious that this is not the case, they said, oh, we've never seen that. We never said that. Well, go and look in the internet and you will say all these famous people, when they unveiled the human genome, that is what they said. So I don't want to go too deep into experiments. They might come later, the way we learn about things like the one you want to know about how cells, why are the cells doing the negotiating, since we have started with that kind of metaphor for how you make a human being. But I hope to have given you some inkling of the situation. Take another very big case. You and I are probably, we are seeing each other in the internet, very different in size. Not very different, but sufficiently different in size. And there is people that are giants, and there is people that are dwarfs, but or less. But the proportions that we have are very well conserved. Our genomes are very similar. So that idea of doing proportions cannot be really simply written in the genes. So take, for example, one of the amazing, marvelous numbers that you can get. Turns out that you are producing, every minute, you are producing 2 million red blood cells. Take that number, 2 million red blood cells you're producing every minute, okay?
Wow.
Your skin is turning over. If you are a young person, it's every month. People like me, it's every 3 months. Turning over. Any change in those proportions, any change in those numbers is going to lead to disease. That's not in the genes. Whatever is controlling those numbers is not simply the genes. The genes are being used, but those numbers are not in the genes. Okay, so these are some examples of things to think about. What is happening? What is going on? We are starting to see that. I mean, I'll give you more examples and we'll discuss more, I'm very sure. But there is no escape from the difficulty of explaining those observations simply on the basis of genes. Now, this doesn't mean that the genes don't have anything to do with it, okay? If you want to do an IKEA kit, you need tools, right? That doesn't mean that the tools don't play a role. They're very important. This is the way I see the genes. They're a toolkit for the cell.
Now, that's fascinating, and I think lots to unpack there in terms of how cells and genes interact, but also what's not in the genes. But before that, maybe we'll take a bit of a step back and talk about your background, because I think it'll be useful to lay context, because you, in fact, were trained as a geneticist and were a professor in the Department of Genetics at Cambridge, University of Cambridge, for many years. But then you realized that we have far overstretched what we explain with genes, and you maybe changed your mind, or I guess just established a better view of biology. When and how did that happen?
Well, first of all, a little thing, because I never emphasized it. I did my PhD in the Department of Biophysics in the University of Chicago. So I was always interested in how could we bridge the gap between the physical sciences and the biology. But I think in the late 70s, which is when I was at the University of Chicago, I realized that was very early. I mean, I failed completely at today that's the relationship between physics and biology is very important, and is very important for this field that we are interested in and we'll talk about. Then I just started working in genetics, and indeed I ended up in the Department of Genetics at the University of Cambridge, first in the Department of Zoology, then in genetics, and I worked a lot on the genetics of the fruit fly. In fact, using genetics to try to understand how organisms are made. And in the late 80s and the 90s, I think there were two darlings of organisms that we could use to understand this process of how you make an organism. One was Drosophila, the fruit fly. The other was the worm, C. elegans. And they produce a treasure trove of genes. Okay, what we were seeing, and it's that if you destroy a gene, you get an animal that lacks a head or lacks legs, or gets a leg instead of a head, or gets a... What was very intriguing is that these genes were conserved. By that I mean that when we look at humans, you don't find that we have different genes, we have the same genes, and in fact, we don't have many more genes. This was a disappointment to many people that thought that there would be some kind of linear relationship between complexity and genes. But you can see the problem here, you remove or disturb a gene, and now you get a defect in the construction of the animal. That's fine. You know what the consequences of destroying the gene are, but what does that gene do? We want to use the terminology. That's much more difficult to find out, because the very early days of genetics were built on the relationship between certain mutations and certain diseases, like thalassemia or sickle cell anemia or alkaptonuria. So there is very simple, because the disease is caused by the lack of an enzyme. The gene codes for a protein that is an enzyme. So there is a very simple relationship. But that's like saying that in these kits that we could talk about from IKEA, or that you used to build radios or whatever, in the old days, today you don't build radios. But, the gene would code for a screw or for a bolt or for a nut, so that's very easy. But when you are in the situation when you remove a gene and a process like making a lens goes by, and you remove this gene and you realize that, or you remove another gene and the heart disappears and the head disappears, it's very difficult then. We are away from the... and that those genes are conserved. In fact, things become even more bizarre. You can take a gene for a human. So this was very, very, very well exemplified by the a gene called PAC6, which happens to be involved in humans. When you mutate that, you get problems in the making of the eyes. There is a similar gene with another name where geneticists tend to be very confusing about this. The gene is the same, has a different gene name called Hyles. And in the fly, if you mutate this gene, you don't have an eye. You can put now the human gene in the fly that lacks this eyeless gene. And now it will compensate for that, but it will not make a human eye. It will make a fly eye. So to me, there are several examples like this. So this means that the genes are tools that are interpreted in a context, and that context has to be a cellular context. So it was in the process of trying to understand what is the relationship between the mutation, that is to say, the defect in a gene, and the phenotype. That's what people in genetics would call the relationship between the genotype and the phenotype. Even in Drosophila, which is a system that is very genetically deterministic, there is a very simple relationship between the phenotype and the genotype. Even there were many problems for me to try to understand. As I say, the catch, and maybe that goes back to the question that you have that you asked at the beginning, how come it has become part of our culture, is because there is indeed a lot of diseases that we can relate to a single gene. Those diseases tend to be associated with failures in enzymes and in very, very different things. But actually, they are a minority compared to the number of diseases that thing.
So going with the IKEA analogy, you're saying if you're trying to get pieces of a bookshelf, the genome is these pieces, and the cells are the ones that are constructing the pieces to make the bookshelf.
That is my view, absolutely. And now you might ask me, how do they do that? I mean, this is the thing. And this is something that we are learning, because as I said, what we are learning is that cells know how to count. They can measure space. They can measure geometry. They sense how many there are. And we are still learning how to do that. But we know that they are very sensitive to these variables, and that the response of the genome to this variable differs a great deal, right? So I think that this is. I mean, we are starting to see that these variables are important. We are trying to understand the language of the cells. This is the thing that is important. Which, of course, the language are words that are encoded in the genome. But back again, there is this relationship, which is not just one way. It's not that the genes govern everything or anything. I mean, for example, take twins. Twins, in a way, are a very good example of what you would expect if the genes were determining who we are, what we are, and everything. All right, so twins, as you know, they have an identical genome, because in fact they result from the splitting of an embryo very early on that should have given at one person, and now it gives rise to two. Watch it. That's also very interesting. Even the earliest stages of development are very, very plastic in a way that are not deterministic. Now, that is very good. So what people working with twins do, trying to understand how much of our being or any part of our persona or as an organ is dependent on gene, all you have to do is to compare twins in many, many traits. This has been done for 150 years. And the fact is, doesn't matter how you take it, things like height, they use the correlation between a character and the presence or absence of that character in the twins is called concordance. And concordance tells you, concordance is actually if you compare twins that are fraternal twins with non-fraternal twins. That's the important thing to measure to get concordance. Something like height is not 100%. In fraternal twins, the concordance is 80%. So even something like height is not. When you get to diseases, and there have been many studies for this, the concordance tend to be 30%, 40% at the best of cases. So that is clearly telling us that there are many events that are not controlled by the genes. Now, some people could say, well, there is a lot of randomness in life, in the activity of genes. Yeah, but I think when you put everything in context, It's an argument that we are building here. It's not one particular statement that is one particular fact that is going to solve everything. It's very, very clear that the genes cannot explain everything. That doesn't mean that the genes don't do anything. I want to stress that. It's just that there are other things that we don't understand. And I think focusing on genes has misled sometimes research, pharmaceutical industry, and it still does in many ways. I mean, it makes money. I think this is in the end a very important thing of the gene-centric view of the world. It makes money, and money drives the world. I think this is going to change. I mean, I'm pretty sure.
So in the case of identical twins, though, they share the same DNA, but you say they have different different features or proportions, or height is different. But most people attribute that to what they call nurture or epigenetics. What do you say to that?
So I think nurture and epigenetics, they are related, but there are two things. I think epigenetics is a deus ex machina that you call when you cannot explain genetics and you don't want to think about something else. In fact, in many ways, the epigenetics, as the word says, is the genetics of genetics. So there is no question. I like to think of epigenetics, which I hope some of the people that are listening know what it is. It's a complex code of the proteins and the DNA that is not transmitted from one generation to another in principle, although we could discuss that. I think that's the mediator between the environment, if you wish, and the genome at the very, very low level. But again, it's a relay. It's a relay. It's not a cause. The cell has to read. I mean, that nurture is imprinted in the genome through the epigenetics. But what is the reading? What is reading that nurture? This is what I think. So, but you're absolutely right. This reliance on genetics that we have to always focus on the genome. You know? I think that's something also very important to understand. And I know that this show has a lot of interviews on longevity. You know, over the years we have heard, well, is it telomeres? Is it diet? Is it this? Is it? No, there is no one thing. I mean, we've been around for millions of years, you know. So therefore, if we relied on one thing to remain on this earth, we wouldn't be here. It's an intertwining of things that have found the balance between them, right? And I think the mediator, the negotiator, since we've started with that, maybe I should have called the book The Great Negotiator, that would be rather than The Master Builder. I think is the cell, because it's the one that can read what's happening outside, be it the environment of cells, and then processing that information, go to the genome and use the genome to build itself.
And you'd mentioned you're still figuring out how cells are reading some of these signals, or they're controlling genes. And some of the things you mentioned in the book is the importance of signaling pathways. Can you touch upon that in terms of how are these pathways being generated, and how are cells using these pathways to, say, control the genes or read some of the signals around them?
So at a very big broad strokes, right? So in the cell there are the genes. There is this process called transcription, which is the process whereby genes are expressed in ways that will make proteins. And then those proteins, there are many kinds of proteins. So there are proteins that are dedicated to this process of gene expression. There is proteins that are dedicated to replicate the DNA. There are proteins that form the structure of the cells, that create a skeleton. But then there are other proteins that are signals that the cell use, sort of words, if you wish, that the cell use. And there are proteins that configure systems that will be able to listen to those signals and transmit them. There are not many. There are not an infinite number. There is a very, very small number. But it's very clear that that's the case. Now, there are two kinds of signals. That is also becoming very clear. Some are chemical, and we know that, and those are encoded in these proteins, in the genes that make these proteins. But then there are also mechanical signals, if you wish, which are the ones that arise from the density of cells, from the geometry of cells, from the number of cells, from the environment. I think at the moment we know a great deal of the chemical communication between cells. We have a very deep knowledge of that. we are not sure how the cells build sentences. We know about the words, but we are not sure how they build sentences. But we know less about the mechanical signals. This is now a field that is blossoming, that it's called mechanobiology. I think for many years people have suspected that cells were able to read mechanics, and that mechanics was a very important part of the biology of making organisms, also of single cells, but after all. And I think this is probably one of the most exciting fields in cell and developmental biology. One of the things that I had a discussion with a colleague recently, because if you think about cell biology, the term cell usually is associated with cell biology or with pathology. That's an interesting dichotomy. And the question is, if we know, and we know a huge amount of the biology of one cell, this is what courses and textbooks in cell biology are about. However, it is becoming clear that when you have ensembles of cells, when cells come together into groups, new properties appear that do not exist in the single cell. In fact, we are just starting to be able to map those properties. Is what I told you, the ability to respond to densities, to create structures, the ability to know when to stop growing. I mean, this is a thing. Your arms don't grow forever. They stop growing at some point. We don't know. People have been pursuing these questions for years. I think now we are starting to and cell biology, the cell biology that we know up to now is not enough to actually answer those questions. So I think there are those two kinds of questions. One of the things I always do, one field that I'm rather involved in, is this trying to build embryos from stem cells, and there is people building organoids from cells. We are not very good at that. And as I say, the way we are doing this is using the same signals that the cells talk to each other with. So we are using their vocabulary, but we're not very good at it because they don't hear us very well. I mean, what comes out is not really what we want, it's what they understand, which tells me that we are not really very good at speaking their language still. Okay, so I think these signals are how do you integrate the chemical and the mechanical signals are very, very. It's quite an interesting thing. I mean, there is people, and I'm sure you might have heard, that talk also about electricity. Well, we can talk about that too, but I think at the moment there are areas where I mean, of course, the nervous system works through electricity. All cells have some electric potential. Electricity is another force, is another physical force, like mechanics that is playing a role. There we are even at an earlier stage, but there is no question that that's also important.
So I think one thing you said, which is super interesting, is that we know how single cells behave, but we don't know how groups of cells behave, how cells come together and form structures and organs and tissues and do all these things, right? And you mentioned electricity as well. I know Michael Levin calls this the collective intelligence of cells, when groups of cells are coming together. And he says the medium of exchange is electricity, or these bioelectric signals. You're familiar with his work. What do you think about that concept? Where do you agree? Where do you disagree?
I think, Michael, it's an effective communicator. I think his ideas are so general that, I mean, when you dig deep, I'm basically a very strong believer in experiments. What he's saying is nothing new. I think maybe he is overstating it, but the proof of the pudding is in the eating. And I think what I'm interested is in the experiments, and I'm not convinced. So some of the statements that he makes, I don't find them in his experiments. I think a lot of the experiments lack rigor, and I think he overinterprets a lot of what he says. This doesn't mean that what he says about the role that electricity might play is not true, but many people are saying that, except that those that work in the field, instead of making these very large statements, they focus on small things. After all, before Darwin explains to us how evolution works through natural selection, there is a whole bunch of people that had pointed out that things could change over large courses of time. The point of Darwin was to find a mechanism for that, and being precise and being with examples. And I'm always finding Michael quite entertaining, and quite stimulating for people that don't know about the field. I find using words like the collective intelligence of the cells. I don't know what that means. He uses terms like agency. Again, I don't know what that means. I think it's all very attractive. It's a very good collection of words. But I think I could develop a similar set of words, and I feel uncomfortable when I find myself talking sometimes in things that I don't know and that I can only represent with words. Somebody said the other day, I heard, I liked it. Words are the clothes that ideas get before they go out in the street. And I think I like that very much, that idea. So I think Michael is very stimulating for other people, but I think that the work is being done somewhere else. And I certainly think that it's another element. It's like metabolism. Metabolism is very, very important. So I could say that metabolism is the collective idea of the cell to produce it. The intelligence needs fuel. So it's the collective fuel of the cell to provide the collective intelligence. So I think very, very entertaining, very, very good communicator. But I think from the scientific point of view, there is a very long way from what he says to, for me, seeing the details.
Do you think bioelectricity is not as important of a signal, or do you think some of the experiments just haven't been replicated?
No, the experiments haven't been replicated, that I can assure you. But when I looked into them, they are so coarse that it's very difficult to come out. Usually the numbers are low. They are over-interpreted. That doesn't mean that electricity, of course, it is a component. But I think it's probably integrated. In fact, I had been thinking I would say probably it's a subset of the mechanical forces of the cell. I mean, in physics textbook, electricity is linked in some way to mechanics. I think I'm more interested in the role that metabolism plays. I used to think that there is only mechanical and chemical signals, but it's very clear that metabolism now seems to be an integrator. Before we came on the air, you mentioned the mitochondria. It's an extremely intriguing organelle. We've thought of it as a producer of energy, and we've been this without problem. And all of a sudden, it appears to be a clock of the age of the cell. It seems to be, and we have no idea. So electricity at this moment, as I say, if you go to developmental or cell biologists, there is some working on very precise things that it's giving returns. I think it will be another element. But in order to find out how it works and where its place is, we need to do better experiments than what Michael has been doing at the moment.
Sounds good. Okay. What about mechanical signals and other cues and chemical signals? I know you said the work is still nascent and ongoing, but do we have any experiments in that realm that's showing how mechanical signals are affecting cells?
Well, no, experiments, there are many. The question is, what are the messengers, the interactions, that the mechanism of those interactions? So, back to electricity, we know how an action potential works. We know how a neuron works. Probably neurobiology is the most advanced of all the biological sciences, to the point that people claim that they can read our minds these days if they set themselves to it. I mean, that's not a joke. I mean, we could do that. But I think that so it's about the details of how electricity works in the other realms, or so we know that something is happening, but we need to find there are some receptors called piezoreceptors, for example, that are able to read changes in tension and changes. There are proteins that are they keep on appearing because it's this thing of genetics. Genetics makes mutants, and mutants leads you to genes. And every time you do an experiment in which you are looking for mutations, things that when you disrupt them will affect the mechanical communication of the cells, a very small set of proteins or of genes come up, but we still don't know how to put them together in a manner that is consistent. I think one of the big problems with this area is that biology, I think, has been very reductionistic for a very long time. And on this, I will agree with Michael that you have to be a little bit more holistic. As I say, what I don't like is the way he's going about it, but because I think you have to be a little bit more focused and a little bit more. And I think the key question, the key term in all this process is emergence. And emergence is a very thing that physicists argue about, and it's very interesting. And it's when you combine two things, and the outcome of that combination cannot be predicted from either of the parts or the sum of the two parts. In the book, I put a very silly example, which I think is true. It's 3 and 3. If you add them, you get 6. If you multiply them, you get 9. If you divide them, you get 1. I mean, and in fact, so that those would be very simple phenomena of emergence. I mean, this is also many people don't understand the difference between a complicated thing and a complex thing. A complicated system is something that can be incredibly complicated, like an airplane, like a car, but it has a blueprint. And if something goes wrong, you go back to the blueprint and you can predict that every time you change this piece, the system is going to respond in a particular way. In a complex system, you cannot do that. What is interesting is that in the course of evolution, nature has found a way to build complex systems that are very reproducible. And we don't understand yet how those systems are kept in that space where they are so reproducible. I mean, we were talking before about the blood or the skin or the intestine, which also has a particular turnover. So, and any deviation from that turnover is gonna create a problem for the organism. So those are emergent properties from the interactions between cells. A cell is an emergent system from the proteins, carbohydrates, and lipids that make it. It's not written in the genome. Interactions between cells create organisms which are emergent properties from this.
And by emergent,
so emergence, understanding emergence in biology is absolutely crucial for understanding biology.
Here you mean that you cannot predict what the outcome would be from the inputs that are going in.
It's that is not the sum of those inputs. It's not the sum of those inputs. So, for example, these days we know so much about cell biology. We were talking about that before, and I'm going to give you an example, which I think is great progress over the last few years. So people are able to simulate cell division, the way a cell divides in the computer. Now, you need that because you know that you are integrating many pieces, and that the interactions, that's an emergent, the structure and the behavior of that spindle and of those is an emergent system. And in fact, when you put all the inputs, you have to model that. And the kind of rules that you put in there are absolutely typical of emergent systems. And you have to let the computer do it. You cannot predict it. And you change a parameter very, very little, and the whole system changes very, very dramatically, which begs the question of how does evolution manage to keep these parameters over the very narrow range that allows life to be the way it is.
That is fascinating. Right. How does evolution make it so reproducible that it's most of us?
Well, evolution. I don't want to give any intelligence to evolution, but is the system. I don't want to fall into these things I was criticizing before of mysticism. No, it's just emergence is a reality of physical systems, and biological systems are physical systems. We just need to understand and wrestle with it.
So you've mentioned this a few times, biophysics, how you got a PhD in that, biological systems are physical systems. Now, this, again, is not a very popular, I wouldn't even say popular opinion, just a common opinion, right? People segregate between the different fields. Biology is separate, physics is separate. Why is biophysics important as a field, and why do you say biological systems are physical systems?
Well, the second thing for me is obvious, but I will try to explain it. In fact, a lot of my interest in this interface came in my undergraduate, and then this is why I went into biophysics, because one of the things that physicists have always been interested about biology, particularly since a very famous book by Erwin Schrödinger called What's Life, is that whereas most physical systems tend to decay into a state of maximum entropy, biological systems are able to build structures to apparently contradict the second principle of thermodynamics. Then a group of physicists realized that there is a set of structures that are called dissipative structures, which mean that very far from thermodynamic equilibrium, you can actually, for a little bit of time, and as long as over time you can balance everything, to do a basically energetic budget, you can, for a very short amount of time, build something of a structure of some sort. So the idea was that this was basically biology is a non-equilibrium structure. It's dissipating energy all the time. That really was for me fascinating. And except that so when you look at life from that perspective, there is a discipline in physics called statistical mechanics. And really, statistical mechanics, one of the most difficult parts of statistical mechanics is precisely non-equilibrium statistical mechanics, which is deals with dissipative structures. And I always say the physicists are very afraid of this because it's very, very difficult. And that's where biology is. So I like to say that it's the unwritten chapter of statistical mechanics. So it is obvious that we are made of. Now, however, living systems clearly have a number of properties which now are coming to the fore, because first we needed to understand the structure of living matter, and that's we've done over the last 50, 60 years. And now when we are trying to put it together, we realize that a lot of what the cells use is, if you wish, physics in a very different manner. Some physicists in the past have thought that you could find new physics in living systems. I wouldn't go as far as that, but it's very interesting that the problems that now that we know the elements, when we try to put them together, we need to resort to physics. In fact, I would say that over the next few years, I think the boundary is blurring between physics and biology, but there is a lot of physicists coming into biology at the moment, and they are the ones that are making great insights, because if you want to understand the mechanics, you need to understand physics. If you want to understand electricity, you need to understand physics. Microscopy has been driving biology over the last 20, 30 years in a big way because physicists are coming into biology. So the relationship between the two, it's obvious. I regret that my view of this was in 50 years ago. I wish I was a graduate student now because I could have a lot of fun. But in the meantime, at least I have learned what are the questions and what are the systems. So there is no question. And as I say, right now is one of the most exciting and thriving fields, because every time, that's the other thing, biologists are used to doing one experiment, getting a result, you make a mutant, you see, you interpret. But right now we have to see the system, as I like to say, the cell, the organism, the embryo, is integrating many different variables into one. So we need, the physicists are very good. We need to develop models that are physical models that allow us to see whether, when we integrate that, what are the relationships between the parts that give us the organism. Physics is very good at that, and that's why we have to learn that.
That's fascinating. I have a question on cancer. You say that cancer is when the genes are rebelling against the cells. But to me, when I hear about your cell's view of biology, I think that, well, cancer is when cells are losing control. So why do you say it's a disease of the genes instead of a disease of the cells?
I have, since I published the book, I've had some discussions on this, which maybe are leading me to modify the part. I'm very glad that this is a very small part of the book. I think that this, I'm not an expert in cancer, and I'm not going to be lecturing or doing. Cancer is a field that is also undergoing a change at the moment. I think perhaps it is that and evolutionary biology. We can talk about Dawkins later if you want to, about the selfish gene. I think it's been the place where this gene-centric view of the world has become very, very important, because, again, we've been able to associate mutations in particular genes with particular groups of particular types of cancer. However, it is becoming clear that view, the more you peddle that view, the more they look into it, the more it's becoming clear that, yes, the genome becomes deregulated in a cancerous cell. But again, for some reason, is not the explanation. So I think you've said it in a way that I would probably put it in the book. I can't remember. The cell loses control over the genome. I think this is true in a cancer, but now, maybe the genome is throwing all these tools out there, which the cell doesn't know how to use and is using in a mad manner. That would be, so there is no question that there is, again, a genetic basis. It's an extreme form this balance between the cell and the gene. I mean, that during development, the cell is indeed controlling the gene. In cancer, it loses that control. And a way of taking forward the idea that I put in the book and that you've expressed here very briefly would be that, it is as if somebody comes when you're building your IKEA thing and starts removing bits and starts giving you these random pieces, and you still have the tools, and it's telling you, do something with that. You're going to end up with something very weird, right? So I think it is becoming clear very slowly that particularly also because although a lot of what they call genomic medicine, it's meant great progress in the therapies of the disease. It's not everything. I mean, last year James Tabery from the University of Utah published a very important book called The Tyranny of the Gene, which addresses precisely the relationship between precision medicine, genetics, political and financial interests, and how we have to be very, very careful with that. We are back into what I mentioned in the beginning. The gene-centric view of the world provides a very palatable, simple, understandable explanation for many things, that it doesn't matter how right it is. Notice that I don't say that is wrong, how right it is, but it produces a lot of business, and that's important. I mean, one example about genes, if I may, it's that, for example, these companies are making money telling you how much DNA you have from Beethoven or from Gengis Khan and people don't realize one very interesting and fascinating thing to me. One thing is genetic ancestry, and the other is genealogical ancestry. It is very likely that you have zero DNA of most of your great-great-grandparents. That doesn't mean that they are not your great-great-grandparents. You might have a lot of the DNA from just two of your great-great-grandparents. Now, that doesn't remove them from your lineage. And that is, to me, another very good example contradicting this view that the DNA has so much to do with us. But again, that makes money. And again, doesn't mean that doesn't have anything to do with us. But this difference between genealogical ancestry and genetic ancestry is one that many people are not aware of.
What is the difference? Can you talk about what
it's very simple. I mean,
is?
Genealogical ancestry, as I say, you come from your great-great-great-grandparents, okay? That's in you people. However, you might have no DNA of most of those. You might have only DNA from two of them. Now, we are told all the time that you can build your ancestry from the DNA, right? By that criterion, you should have inherited DNA from every one of your great-great-great-grandparents. Mm-hmm. Now, that's not the case in many instances. That doesn't mean that those are not your great-great-great-grandparents.
Right. So that research is wrong then.
No, it's not wrong. No, they haven't told you that. That doesn't mean that it's wrong. It is the belief. It is the belief that we believe. I mean, you can identify your parents, probably, yes. Your grandparents, possible. Your great-grandparents, the minute you go back, you start the lineage, because the DNA is not inherited in a 50-50 basis. This is the point.
Fascinating. Okay. So let's talk about Dawkins and selfish gene. Why is the selfish gene theory incorrect?
Again, I think it is important. The headline is Dawkins is wrong. I think it's incomplete. I think that delves into some of the things that have been discussing. Dawkins' view of Dawkins, with his book The Selfish Gene, has done more than anybody else to popularize this idea of the gene as a central element of our being, of our lives. And I think the idea is based on two statements, if you wish. One, very biological in a way, that it's the ultimate replicator. Basically, the gene, it's the thing that, as he said, wait a million years and you won't have the organism, but you will have the gene, okay? Because the gene keeps on being replicated and passing on. And the second thing that he said, which as a student, when I read the book, I was fascinated by this because I was interested in biology, is that we are just machines that genes build in order to pass themselves on over periods of time. Dawkins is what it's called in the trade a behavioral ecologist. He's interested in the fights that different animals have with themselves for survival. It's a branch of Darwin. He knows nothing about embryos. He knows nothing about cells. And he obviously hasn't realized that in order for a gene to build an organism, it has to make a cell. And then we are in the situation where we are. But he also doesn't know that there are other replicators besides the gene in the cell. Take a cell, take the membranes of a cell. You cannot create those out of the DNA. You can put DNA in a tube with everything you want. Membranes will not appear. And the cell is a very complex system of membranes. In fact, the membranes are replicators as much as the DNA, and they are totally independent of the DNA. If you wish, I mean, that's why a virus likes to infect the cell, because in that environment it can have many copies of, but without the cell, the virus is nothing. So we already have the idea that there are other replicators, okay? Then the other thing that it's interesting that he says is that only the genes passed on during generations and go on forever. Well, I have news for him and for anybody that is interested in this. But I want to stress, this doesn't mean that he's wrong. It's just that we have to complete the picture that is drawn for us with the ideas that he's given us. And it is the following. It is that germ cells go on to infinity. Germ cells are the cells that are going to give rise to the gametes, that are going to give rise to the sperm and the egg. Very early in development, before you start building the body, the system separates few cells that they are the germ cells. That's where this DNA, they are the vessels for this DNA that is going to pass on. In fact, these cells are so special. They are going to be passed on to the next generation, and they will be set apart again in the next generation. So they travel with the DNA. They are the vessels of the DNA over generations. So it's not just the DNA that is passed on. The germ cells are immortal in many ways. He's never thought or discussed the implications of this. I mean, my view about this, and I think this is what I explain in the book, I think the cells and the genes make a Faustian pact, like in the book Faust. I mean, the genome says to the cell, well, you can do with me whatever you want during development. You can use me as long as you provide me a little traveling ship to go into eternity. And those are there you cannot touch me. But during the development of an organism, in the short term, you can do whatever you want. So I think that's the thing. Then when we go to the second statement, the organisms are just designed by, well, we've discussed that in the last hour, that's clearly not the case. Maybe the genome builds the first two cells, because when you get the two gametes coming together, they have to negotiate, the two genomes have to negotiate quite a number of things, because they are very different in the way the chemical construction that they have. But once they generate two cells and cells start talking to each other, you have a totally different parameter here. Okay? Once you get the first cell, as I say, maybe the genome has a lot to do in sorting itself out for that first cell and to get the first cell division underway. But once you have two cells and they start talking to each other, that's a totally different business. So I think that's why I think it's, even from the perspective of a gene, Dawkins tends to see it as a number, something, an entity, not as a molecular entity that makes proteins and that is embedded in the cells. I think for him, it's an appealing idea. It's simple enough to catch the eyes of people. It has sociological implications. I like to think that cells, the genes might be selfish. Cells are altruistic. They collaborate with each other. They build organisms. They build beautiful things. And that, we don't know how these instructions are preserved, because we are just learning the language of cells, as I said. And they use genes. They have a very constructive relationship with the genes, but they're also very altruistic in the way they cooperate and they build. So I like to contrast the selfish gene with the altruistic cell, and maybe in the future we will learn. Life is a combination of selfish and altruist, but the altruists always win because they are the most creative.
But perhaps taking the other side, you could make the argument that, sure, the genes need the germ cells to be transported. But the genes are so clever here that they're dictating the cells on what to do, and they're dictating cells on how they want them to behave. And that, I think that's what Dawkins is trying to get at, where it's like the gene is always trying to replicate, and maybe it's not the cells controlling the genes. Maybe it's the genes
no. So
dictating
the cells. No, well, I think it's a pact. I think it's a pact because a germ cell will be later used to start building an organism and set itself apart. I mean, who is running what in that cell? We don't know, okay? We don't know at the moment, because I think the first decision, it results also from certain interactions. They have to make a partition. The fertilization of the egg creates a set of cells that are fairly naive, and then these cells, in fact, in all organisms, the germ cells are set aside before any decision about what the cells are going to do. So the cells are being very naive and a decision is made. You could tell me the genes make that decision. Well, at a cost, because they are going to allow the cells to be a lot of crazy things with them in the building of the organism, okay? I think this opens up an interesting area of research for both evolutionary biologists, philosophers, if Mike Levin wants to contribute to this. I think it's an interesting connection. How much of the development of the organism is determined by genes? Okay, I think there are organisms in which there is a lot of that, but in vertebrates, in mammals, I think the cells are running. I'm leaving aside the issue of the germ cells, where I think you have posed an interesting question there, but I think that. That reminds me a little bit about an anecdote of Glenn Gould and Leonard Bernstein when they were going to play together, and Bernstein asked who is running. They were going to play a piano concerto of Brahms. Who is running this, the conductor or the soloist? So you could imagine that the soloist is the DNA, and the conductor is the cell with the whole orchestra. So who is running what? That's what happens in the germ cells. But in the development of the organism, I think the more you think these features that we have discussed, the scaling, the homeostasis of the system, the knowing where to stop growing, the turning over of the tissues, very difficult to see how that is written in the genome. Notice that I say it's very difficult to see, but it's not gonna. I'm not gonna become, oh well, there is a gene, and when you no, I think, and as I say, we are now starting to see that when cells come together, they develop properties, particularly when we are finding these remarkable possibilities of building embryos or of building organs. That's not done. In fact, we can do that without playing with the genes at all. It's simply playing with the language of the cells, which tells me that there is a language that we need to understand.
Right. I do think that's a good distinction. Maybe we need to look at germ cells separately and study them versus looking at development. So, putting it all together, you said Dawkins' theory is not necessarily wrong, it's incomplete. How would you complete it?
Oh, watch it. If Dawkins was here, he would say, I don't have a theory. I have just put together an idea, and Dawkins will always say that, after all, Dawkins is Darwin. It's the prophet of Darwin. Not the prophet, he's the preacher of Darwin. It's interesting because, as you know, Dawkins had a very big running argument with Stephen Jay Gould, who is the other great evolution popularizer. It's interesting to me that I think Gould was much more holistic than Dawkins, and Gould did appreciate the role of development in understanding evolution. He was never able to bring it together in a manner that would make sense, but he had a sense that was important. Whereas this very extreme gincestry of Dawkins, I think the way to complete it is precisely what I'm saying, is to ask questions about how these forms that we call animals and plants arise, not because of the genes, because then, but and how they are selected. What are the forces that are shaping these ensembles of cells, and then how? I also tell people, I think humans, we always think that the time in which we live is the best and is the one that we know most, and I think particularly because we have had these leaps in the 20th century that have put us where we are, we forget that at the beginning of the 20th century, in the context that we are talking about, we didn't know anything about genes. We had a lot of questions, which are the same that we have now, and we know nothing about genes. Then we find genes and we make them an explanation. Watch it. Genetics is a method. I also say this in the book, and we've turned a method into an explanation, which I said, it works, and CRISPR works. There is no question that for certain things it's a very small, but there are many questions. There are very large questions that we have discussed here. Now, it would be foolish of us to say that we can explain everything with the genes, because although we can explain a large amount and use it, that's really the point. When we know something and understand something, we can use it and we can do genes. But there is a whole other area, which I think is exciting from the point of view of young students and of young people. There is a lot of area that we don't know. Maybe in a way, Michael Levin is doing good in talking about electricity the way he does, because he's opening the door to something. And I think we need to dig in there. There is a story that I like to tell about phlogiston. Maybe, I don't know whether you remember or you've heard of phlogiston. Phlogiston was something very important, and I think it's interesting in this context. So for a long time, people were trying to see what happened when you burn substances. This is in the story of alchemy. So they thought that when substances were burned, they released some magical element, which they call phlogiston. The problem is that when they waited something before burning and something after burning, it weighted more. It weighted more than before burning. Therefore, phlogiston had to have a negative molecular weight. Okay? So this made no sense, but they were all very happy with this until they found oxygen. That oxygen was being incorporated. So I think we have to be careful, you see, with building theories or using genes to explain things that they can't explain. And we have to admit that. So I think that this would be my thing, that I think the experiments that we are doing with ensembles of cells, particularly when to try to build organs or embryos, they are telling me, in fact, I mean, this brings us full circle to the beginning. Yes, I was very interested in the connection between physics and biology and remain very interested in that. But since we've started to work with these systems where we make organs, buildings, I don't even know the questions. I don't understand what is that we are seeing. So I think part of what my generation have to do is be very clear to accept that there are things there that we don't understand and highlight them so that then we can find the answer.
I think you're making an important point, because you're saying that, sure, genes explain some things, but they don't explain everything. And what we've done in biology since the discovery of the genes in the 20th century is we try to use genes to explain everything. But then there are all these other mysteries, cells being one, which we don't understand. And clearly they're doing something which genes are not doing. So, go ahead. No, this makes me more curious about the history as well. Before the 20th century, was the cell's eye view of biology more predominant? Was it more in the center?
I think there is no, that's you use the term that I use in the book, is cell eye of biology, because the people that are very Dawkinians, they use the term gene eye of biology. I think the cell's eye of biology, it's warranted, particularly by the last 20 years, where we have seen that the cells are not just bricks that come together, that are put together to build organisms, but are actually sentient, computationally very alive. I mean, there is clearly, if you want to say that there is some kind of an intelligence, I think that's going to match, or an agency. I'm more interested in the mechanics. But I think in the 19th century, they were seen more as blocks, not as agents, as something that had a life of its own. I think now, particularly due to the microscopy, to our understanding of cell biology, to the poking individual cells and to see the differences, we can see that they are something that we still don't understand and that have properties, particularly when they come in groups. So I think I always say the last century, because I am a 20th century person, but I mean 2 centuries ago, when cells were discovered, they were seen as building blocks. I also discussed that in the last part of the book, that I think this is a very new view that is coming. I mean, the cell as a very active element, which comes above a very different level of the genes. Sometimes, if you wish, is like the same way that the physicists try to bring together the different forces from different scales. I think we have a problem in biology, but maybe if you want to study what cells do, you maybe should not worry about what genes do and think about cells. If you want to study what genes do, probably you don't need to worry about cells.
Let's dive deeper into some of your work in development, because I think embryonic development is just fascinating. It seems like magic, and it seems like such a black box. And correct me if I'm wrong, but I think you're trying to understand it, particularly through the use of something called gastruloids. So one, can you explain what is your work in this area? What are gastruloids and what is gastrulation? So a lot of questions at once.
Well, let's go slowly. I think I spent many years working with fruit flies, which is a very gene-driven organism. But there were things that I saw in trying to understand, as I said, how genes control development, if they do, that made me wonder what that would be. There is a very good sentence from Richard Feynman. When he died, he had written in his blackboard these equations, his everything, and he had one sentence that stuck since I heard it. That which I cannot understand, I cannot build. I mean, there are many variations of this. So I thought that is true. If we really think that we understand something, it would be good to be able to build it. And then we can learn. I mean, usually you see in a genetic analysis of a process, we take things apart. We take pieces apart and see the consequences of that. There is a very famous paper called Can a Biologist Fix a Radio? I recommend people to look at it. It's quite, because we are just taking apart things and making copies. But if we could build it, it would be a different approach. Then I heard about embryonic stem cells. This was around the year 2000. And the thing that fascinated me about embryonic stem cells when I heard about them is that in the dish, you could differentiate them. They could become different cells of the different cell types of the organism, but they would not make an organism. However, if you put them into a very early embryo, they would contribute to that embryo, and they will build the organs in very proportion. So that posed the question: what were we missing in the dish? Or there was magic, as you said, because development is magical. So that led me to switch and try to see if I could answer this question, which is what drove me. And working, this was a time when I think stem cells were, from the beginning, they were very much driven towards regenerative medicine, but a very small group of us in the UK thought that maybe we could use it to understand developmental biology. And my question is whether we could use them to build organs. And after many years, one day, after trying many things I saw these structures appear in the dish, which were like an abstraction of an embryo. They were, as is Picasso, as a colleague of mine said, as if Picasso had painted an embryo. And I was very, very, very surprised. They had the 3 axes, the genes were very perfectly organized. So they are a model of the embryo. There were things missing in terms of structures, but the genes were very, very organized. And I thought this is a very, very good system to start understanding how cells are put together. Now, immediately many people might think, well, what did you add? What was the crucial variable, the crucial feature that we have to control is the number of initial cells. And this is what I was telling you about in the beginning. And this is a question that now we and others are working on. Interestingly, the number of cells that we have to put on is about the same number that the embryo has when it starts building an embryo, that the cells of an embryo. So there is something magic, in the sense that scientifically magic is something that is attractive, begs a question, and you have to find the answer. It's a challenge that you see. But it's really dramatically interesting that only we put a very precise number, and you cannot go off by about 50 or 60 cells. If you go over 50 or 60 cells, the system doesn't work perfectly anymore, or doesn't work at all. So I think that the gastruloid, we call it a gastruloid because gastrulation is a process. So when an egg is fertilized, the cells divide a number of times to create a mass of cells. Notice that you don't use the space to divide the space. You create space that is populated by cells. And now at a certain moment, these clusters of cells, and the moment varies from one organism to another, starts a choreography that is called gastrulation. And that my late friend and great biologist Louis Wolpert coined that sentence that the most important moment in your life is not when you're married or when you're born or when you die, is when you gastrulate. Because that is the moment with this choreography of cells, which again, this is very difficult for me to see how that's imprinted in the genes, because this is cells all of a sudden measuring space, knowing where to move. It's a very, in some organisms, it's incredibly complicated choreography. And yet it is reproduced over and over again, and the cells seem to know that. Seem to know when to move, and they're all doing it in relative to each other. Is looking at movies of this, there are increasingly many, is absolutely beautiful. So the outcome of this process is the body plan, is the organism with the head at one end, the tail at the other, the back and the belly, and
we're able to see this. Because this is still an embryo, right, at this point?
Well, this structure, we can see this in embryos. I mean, but when we aggregated the embryonic stem cells and did a little bit of chemistry, a little bit of alchemy, they organized themselves as long as we started with that very precise number of cells. Now they formed something that was very much. Interestingly, everything was organized in a mass of cells. If you looked at it, it would look like a sausage. But when we started looking at the genes, they were perfectly organized as they are. What was interesting, and this is the magic of it, and I'm going to oversimplify here, these things are floating in medium. These things are floating with medium. If we now embed them in a matrix, in some physical things, now morphology starts appearing magically.
Wow.
So this is something that we are still recovering from, if you wish, but it's given us a window and an experimental possibility. And here you can see why I said earlier that we are still trying to define the questions because this is not the product of the activity of one cell. This is clearly something that results from interactions, dialogues, negotiations, to go back to the beginning, between groups of cells.
You said that you have to start with a certain number of cells. That's critical to the experiment.
Yes.
Now, I'm a little confused there because, I mean, the embryo starts with ultimately a single cell, right? And it keeps dividing. So what do you mean by that?
No, the cell first, the embryo starts from one cell. I mean, the term embryo, it's that would lead us into a totally different question about what is an embryo. That is not the topic of this, maybe of another conversation. But the first thing that cell does is to amplify itself, to modulate. Cells are not going to start becoming different from each other until there is a certain mass of cells, okay? So during that proliferation, which is really basically the cells are not growing. In most embryos in the first day or 2 days, they are not growing. They're just dividing that space and making different cells. Then when there is a certain number, they start becoming plastic and becoming creative in space, okay? So, and the number that we have approximates the same number that the embryo, to talk about that structure, has at the time that it starts undergoing those movements and those choreographies.
I see. Okay, interesting. So you've tried it with different numbers and it doesn't seem to work.
Absolutely.
But would it be different would that set number be different for different organisms?
The only other, a colleague of mine, Ben Steventon, has done very similar experiments with fish. Fish is not, because we were in the same group and we collaborated in this, and there is also a minimum number of cells that that has to be present in order for this to happen. But fish is very different, and I wouldn't establish comparisons here without going into detail that will be boring for everybody. But in the other organisms where these structures have been analyzed in detail, there is also a certain number of cells above or below which it doesn't happen. You cannot form a fish-like structure, which would be in that case. So we are revealing a very, very basic properties of groups of cells. In fact, that leads into the question of, during many millions of years, organisms existed as single-celled. Then they became colonial. So sometimes they would come together to defend or to feed. But then there is a big jump into making animals and plants. Those are multicellular, right? So there is a big jump there. What happened? We don't know. In fact, the more we know, it's not just simply a matter of genes, because we find many of the genes that we find in multicellular animals and that we used to think they are associated with multicellular animals, they are also present in unicellular animals. So something happened which probably has to do with these properties that we are revealing in these systems like the gastruloids. So gastruloids at the moment for me are a window into trying to understand how cells build organisms. But then as a side product, and I think there is people actually using them to check on bioelectricity. There is many people checking them in mechanics because you don't have to use animals, you see. And I think that's the very big important thing. They're also telling us many things that, as I just told you, if you don't constrain them mechanically, they organize the genes very well in space, but they don't create shape. That's telling us that shape and gene expression, you see, that's the very clear demonstration that there are two different things that come together into an organism. In fact, as I sometimes tell in talks, it's very fascinating that Alan Turing, that many people here will recognize as the inventor of the computer, the great British genius, he has a paper towards the end of his life before he unfortunately committed suicide, which is called The Chemical Basis of Morphogenesis. It's an absolute classic in developmental biology. And at the beginning, the introduction is a fascinating piece of writing. He makes a very prophetic statement. He says, an organism, I'm paraphrasing here, it's an interesting combination of the chemical and the mechanical. And he says the interesting point, the combination of the two makes the problem very, very difficult. So you should be able to separate it. And now here I'm going to deal with the easy bit, which is the chemical, because it's the mechanics that has the problem. Isn't it fascinating that the gastruloids are telling us that we can separate these two things? So what Turing used as a very physical, theoretical construct, we can see that it is, and that evolution, what has done is to breed the things together. So I think it's these things are being very fascinating for many things. Then they can also be, and there is people working on that, the seeds to generate organs, to then generate certain cell types. So I think I find it very fascinating, this Turing. When I learned about this Turing insight, it was remarkable, because that's what we are seeing. One thing about biology that I believe is that sometimes we are too concerned about causality. So we always think that everything, I think evolution works with many things that brings together into a seamless object, which is what we are. But actually, sometimes there is not really causal relationship between things, it's what works. And that's where emergence also probably plays a role. But that brings us back to what we were talking before. How do you keep these features of animals and plants that are not really simply written in the genes? There is a collaboration, but I think we need to understand cells. I always say the 21st century is the century of the cell. There is no question in my mind to that. The same way that the 20th century was the century of the gene.
I totally think your work on gastruloids is fascinating because it opens up so many avenues for research. So I want to close with asking you, what are some of the questions that are top of mind for you that you want to explore, either be it in the realm of cell biology, gastruloids? I know the two are connected. So what are the top three questions you're exploring?
I think they are very, I think as somebody once said, I'm afraid of asking very big questions because I'm going to get very small answers or no answers. So I prefer I think I'm very, very glad to have been able to create a system that is going to allow us to study these basic properties. I think the questions that we are very interested in, it's these questions of how do cells read the number and use that information to start creating the coordinates and an organism. That's a question. And also, we haven't talked about the intricacies of gastrulation, which are very conserved, and I'm interested in how are they written and why they vary between animals. Very focused on those very early steps and how, but particularly is to define those properties, to define the system, those features of cell ensembles that for which we need to find mechanisms. And what are those mechanisms? I think those are my questions.
Alfonso, in closing, is there anything else you'd like to add?
Well, in the context of what we were saying at the end, as a person with curiosity that have always been interested in understanding how organisms are made, I'm very glad I ran into gastruloids, because to paraphrase Feynman, that which I can build, I cannot understand, which opens up a whole bunch of questions, and that's what science is about. Maybe not about finding answers that close problems, but about finding answers that open new questions. Thank you.
I love that. I think questions beget more questions. That's amazing.
Yeah, thank you.
If you enjoyed the podcast, you might also enjoy my newsletter, livelongerworld.com, where I share practical longevity tips and also upcoming releases of my podcast episodes. So if you wish to subscribe, click on the link below or head over to livelongerworld.com. Thank you for listening, and I will see you next time.